In this session, we will focus on reporting guidelines and standards, and why it is important to know them and keep them in mind when publishing scientific articles or reports. We will split this topic into two parts, and we will first look at the ARRIVE guidelines in more detail as an example for reporting guidelines for in vivo research, and will then, during the second part, have a closer look at other reporting standards, including the minimum information guidelines for different scientific methods and techniques. In general, the aim of reporting standards is to ensure that results can be understood and interpreted properly by any reader of such reports or publications, and that there is sufficient information that results can be reproduced by others and independently verified. If you remember the outcome of the Cancer Reproducibility Project, which we have mentioned in Lesson 1, here scientists involved in the project have tried to reproduce over 109 experiments, but not for a single one they had sufficient information to understand and follow what the authors from the original publication exactly did. And that's a situation which is really not acceptable. In this context, it is also important to mention that when we write protocols for our experiments, that we actually define in these protocols which information and experimental factors we want to document and/or monitor, as this has direct consequences for when we later summarize and publish our results. Information which we haven't collected during the experiment might be lost and might not longer be available when we report about the results generated. As this is an important aspect for high-quality reporting, we will come back to this point later during the lesson. Let's first look at the evidence that reporting guidelines are actually needed. One important basis for developing new guidelines for the reporting of in vivo research was the paper by Kilkenny and colleagues. Here, in 2009, the authors analyzed 271 in vivo research publications to see whether critical information regarding quality measures were actually reported. Overall, measures against bias were rarely described in less than 20% of publications, as you can see here for the reporting of blinding on the left side and randomization on the right side. Importantly, incomplete reporting does not allow us to say with 100% certainty that these protective measures have not been implemented. But it makes it very difficult for us as a reader to understand the quality of the studies described in these publications and to judge how robust and reliable the reported findings are. And because of this widespread incomplete reporting of critical information, a working group of experts from different interest groups came together, including journal editors, funders, statisticians, animal welfare experts, and scientists, of course. And they thought about how reporting of in vivo animal research could be improved so that any results and research outcomes can be used to their maximum potential. Following these discussions, the working group published their results as the so-called ARRIVE guidelines, which is short for Animal Research Reporting of in Vivo Experiments. The most recent version of the ARRIVE guidelines was published in 2020 and contains just over 20 items, which should be considered to describe a study comprehensively and transparently, and to allow readers to judge the internal validity of a study and to be able to reproduce it. And besides having access to sufficient information to understand and reproduce findings, one additional benefit of aligning on reporting standards is that it helps to generate high-quality systematic reviews where certain pieces of information from multiple publications are summarized to combine and compare the findings of many different studies or research outcomes. Basically, the ARRIVE guidelines contain two sets of items. A list of 10 essential items contains the most critical items which really must be addressed. The essential 10 describe information that is the basic minimum to include in a manuscript, as without this information, reviewers and readers cannot confidentially assess the reliability of the findings presented. It includes details on the study design, the sample size, measures to reduce subjective bias, outcome measures, statistical methods, the animals, experimental procedures, and results. The second group, referred to as the recommended set, adds context to the study described. This includes the ethical statement, declaration of interest, protocol registration, and data access, as well as more detailed information on the methodology, such as animal housing, husbandry, care, and monitoring. Items on the abstract, background, objectives, interpretation, and translatability also describe what to include in the more narrative parts of a manuscript. Together with the essential 10, the recommended set represents best reporting practice. The ARRIVE working group also published additional explanations and justifications for all selected items. For example, they explain what evidence do we actually have that a certain item is indeed critical. They provide definitions and clarifications for technical terms and present examples from the literature of how it could look like when using and adopting the ARRIVE guidelines. We will now look at a few examples. For example, item 3 of the ARRIVE guidelines asks for defining inclusion and exclusion criteria before starting the experiment. And this is a nice example from a real paper how you can easily achieve and describe this in your paper. So animals were included in the study if they underwent successful MCA occlusion. And animals were excluded if insertion of the thread resulted in perforation of the vessel wall, and so on. So it is very clear for the reader to understand what happened, and it is quite simple to do when writing a manuscript. Another example is item 7, asking to specify what the experimental unit actually is in the experiment. And again, here ARRIVE provides details that, within one report, the experimental unit can be defined differently for different studies. And in one case, the single animal was considered the experimental unit. And for a second set of data, the example provided describes the litter as the experimental unit. Again, very clear for the reader to follow what happened, what sample size meant in these studies, and what data were subjected to statistical testing. The third example is about item 8 in the ARRIVE guidelines and the need to provide sufficient details about all animals used. And the ARRIVE guidelines specify what is expected, so details like strain and substrain, sex, age, weight, the supplier, and so on. Again, as you can see here in the blue box, it is very simple to make sure that all this critical information is provided for the reader to understand and to potentially reproduce what has been done in the original study. The ARRIVE guidelines had some success over the following years after they had been introduced, meaning that by now over 1,000 journals indeed recommend the use of the ARRIVE guidelines before submitting a manuscript, and more than 40,000 copies were requested and read in over 50 different countries. The main question, however, is of course whether or not scientists do actually use the publication guidelines. So this is a survey from 2016, and the authors ask about 300 scientists from different interest groups whether they have heard about the ARRIVE guidelines and whether they are indeed familiar with them. And as you can see, the majority of survey participants has actually never heard about these guidelines. Another interesting study was published a few years ago addressing the implementation of the ARRIVE guidelines at journal level. The authors compared journals which do and which do not support the ARRIVE guidelines and looked at two different time points. 2009, before the ARRIVE guidelines were published, and 2015, 5 years after they were published. And what you can see is that there's almost no difference between journals supporting and not supporting the guidelines in terms of fully reported ARRIVE items. And there's almost no increase in the percentage of fully reported ARRIVE items between 2009 and 2015, and the slight increase is actually the same whether or not journals supported the guidelines. So that means that adoption of the ARRIVE guidelines, unfortunately, is still quite poor. And that's why it is so important that educational initiatives like this course exists that explain what the ARRIVE guidelines are, why such reporting standards are important, and why a relatively minor effort by us scientists and authors of publications can really make a difference to those who will read and follow our research.